Hello, everyone. My name is Matthew Nguyen. Sorry, one second, I'll adjust this. I am a fourth year neuroscience major, as was explained. My faculty mentor is Dr. Tara Nordgren, and today I'll be presenting my research related to the carcinogenic and pro-inflammatory effects of chronic agricultural dust exposure on lung gene expression. So first, I'll start out by discussing the context for our research, starting off with inflammatory lung diseases. Agricultural workers and farmers are at an increased risk for chronic lung diseases, such as chronic obstructive pulmonary disorder, also known as COPD, which includes emphysema, asthma, and bronchitis. In addition, they're at an increased risk for fibrosis and lung cancer as a result of the chronic inflammation that they're subjected to. A study conducted in Europe found that the prevalence rate of COPD in non-smoking animal farmers was 17.1%. And furthermore, these workers specifically worked in animal confinement facilities. Now, these agriculture and livestock workers can work anywhere from dairy farms, poultry farms, hog confinement facilities, cotton textile mills, and more. However, this list of examples are ones where epidemiological studies have demonstrated these workers specifically to be most at risk. Now, our work specifically revolves around the inflammatory effects induced by dust exposure from hog confinement facilities. So to talk about organic dust content specifically, pictured to the right-hand side, we have an example of a hog confinement facility. But rather than using raw dust, we used an extract of it, which is created by collecting the dust that is settled on the surfaces of these facilities, sterile filtering it, and then diluting it with saline. The contents of the dust include bacterial proteases, peptidoglycan, fungal components, and lipopolysaccharides, also known as LPS. Now, previous studies have demonstrated that acute and repetitive exposure can induce an inflammatory response. However, carcinogenic potential has never been measured in the context of chronic exposure specifically. Now on to discussing the role of inflammation in cancer. Inflammation is a beneficial response activated to restore tissue injury. So unregulated inflammation can become chronic and potentially result in malignant cell transformation in the surrounding tissue. This leads to an increased risk for carcinogenesis. And to provide more context to the pathology of this, cancer is the second leading cause of death in the United States, with approximately 600,000 people dying of it each year, according to the Center for Disease Control. In addition, smoking and secondhand smoke cause about 90% of lung cancer deaths in the US. And furthermore, NNK, which is relevant to our project, is one of the 7,000 plus chemicals that is found in cigarette smoke. Previous research has shown the inflammatory response that dust elicits following exposure. However, no study has yet investigated the carcinogenic and/or tumorigenic potential of the dust following chronic exposure. On to study design. To assess the carcinogenic potential of dust, AJ mice were intranasally exposed to hog dust extract, also referred to as HDE, 3 times a week for 24 weeks. Additionally, a one-time 100 milligram per kilogram intraperitoneal injection of NNK was given at the beginning of week 4. NNK is a tobacco-specific carcinogen that is used to induce tumorigenesis. Additionally, AJ mice have a mutation that makes them particularly susceptible to developing tumors spontaneously. We harvested the tissue of the left lung to use for gene expression, and there were 4 experimental groups that were used for our study. There was one with saline and no NNK, one group with saline and NNK, an HDE no NNK group, and lastly, an HDE with NNK. Moving on, we performed gene expression analysis using NanoString. We ran a pan-cancer mouse immunology panel with 749 genes specifically related to inflammation and cancer. So this allowed for the pathway analysis and transcript level gene expression. And of the 749 possible genes, 218 were found to be altered, and 129 were differentially expressed between the HDE and saline exposed mice. These 129 genes were sorted by p-value, and of those genes, we selected the top 20 highest, sorted by fold change for analysis. So next up, to discuss the differential gene expression, pictured over here on the left-hand side is a table of the top 20 genes sorted by fold change that was mentioned previously. And now all of these are related to inflammation in some capacity, but rather than boring you by going through all of these genes, I'll talk about a few of them. Starting off with CCL6, it is a chemokine ligand that is produced by macrophages, fibroblasts, and T cells, all of which are hallmarks for inflammation and chronic lung disease. It generally acts as a macrophage chemoattractant, which basically means it signals macrophages to come to it. And CCR2 is a chemokine receptor that is responsible for mediating monocyte and macrophage recruitment to sites of inflammation. Lastly, CD14 is a surface antigen that is preferentially expressed on monocytes and macrophages. It cooperates with other proteins to mediate the innate immune response to bacterial lipopolysaccharide, which we did mention earlier is a component of the organic dust. So therefore, upregulation of CD14, for example, is expected because it signifies the exposure to the dust. So now to discuss the significant pathway alterations. On the left-hand side, we have a heat map providing a visual of the multiple pathways that are altered as a result of dust exposure. But furthermore, looking at the top, we can see that there's a clear separation between the saline and dust exposure groups. This is evidence that supports the fact that dust is actually driving these pathway alterations rather than NNK. So if it was mostly responsible to NNK, we'd be observing 4 clear separations up here in the treatment area to represent our 4 experimental groups. But once again, this clear separation is between the saline and dust exposure groups. So to further substantiate the finding that dust is driving these increased pathway alterations, we statistically analyzed the mean Z-score shown in our bar graphs to the right. And what we see here is a statistically significant enhancement of pathway alteration by the dust, not NNK, it's independent of NNK. And it's specifically for the cancer progression and inflammation pathways. Now, when looking at the genes related to the cancer progression pathway, we do also see significant alterations in EMT-related genes. EMT, it stands for epithelial to mesenchymal transition. And this is the phenomenon when epithelial cells begin to lose their rigid structure and become mesenchymal. So taking on a more mesenchymal phenotype is associated with a little bit more fluidity, which is a characteristic associated with tumor metastasis, as you can see in this diagram. And the 3 reasons for why EMT may occur include development, wound healing, and cancer progression and metastasis, which is the most relevant to our project specifically. Hallmarks of EMT include upregulation and downregulation of certain genes, some of which we have identified down below. Now, one hallmark of EMT is the loss of the tight junctions that maintain cell-to-cell adhesion. So it's the loss of these tight junctions that help the mesenchymal fluid phenotype develop. One of the genes that we identified is CDH1, which you can see over here. And CDH1 is a gene that encodes for the protein ecadherin. And ecadherin, as you can see over here, is one of the proteins that's crucial for maintaining this cell-to-cell adhesion for the epithelial cells. So therefore, the downregulation of CDH1 suggests that these cells are possibly going through EMT. So to summarize these findings, overall, we found that gene expression was driven by dust, not NNK. A total of 129 genes were differentially expressed between the HDE and saline exposed mice. HDE drove upregulation of inflammation and cancer progression genes, demonstrating its potency. Included in these genes were ones related to EMT, which is a well-established hallmark of cancer. This study was the first of its kind to analyze the transcript and pathway alterations following chronic HDE exposure from agricultural organic dust. Lastly, we hope that these findings pave way for greater research to be done regarding safety measures needed to be addressed for agricultural workers. And then lastly, I have acknowledgements for the faculty members and graduate students in the Nordgren lab. Thank you.